So, hopefully everyone can hear me while I wander the stage, as is my wont. So the one thing that I would like you to take away from Repair Biotechnologies, talking about this company, is that we can reverse atherosclerosis. And that's an important thing, as I normally don't speak right after another company that can also try to reverse atherosclerosis. But we'll use this to our advantage here, so I don't have to repeat myself a lot. So we'll skip over the boring stuff. So the important thing about atherosclerosis is that it kills a lot of people. It is the biggest problem for humanity in terms of people dying and having things that lead to dying. So look, I don't have to repeat much of what Oki said there, but you get fatty plaques. They grow in your arteries. We don't have to talk much about why this happens right now, but they obstruct the flow of blood, which leads to heart failure, vascular dementia, a whole bunch of other things, because blood's really useful. It has to go places, and if it isn't going places, you suffer. But more pertinently, if this plaque ruptures, if it's a soft plaque, if it's a fatty plaque, it ruptures, debris goes downstream, it blocks an artery for a while, and that causes 27% of human mortality between stroke and a heart attack. You can choose a better lifestyle, you can take lipid-lowering therapies, but these cannot reliably reverse existing plaque. They can only slow it. You're looking at sort of a 20% reduction in mortality risk by adopting on average, by adopting the best strategies. But we have a therapy that can reverse plaque, and that is the big deal about what we do. So, plaque, it's a bad thing. If you look at the risks, the hazard ratios on the right side, a person with no plaque here has this many cardiovascular events. Now the plaques in this study, this was an imaging study done in 2004, and they went and imaged just major plaques in major arteries. So if you have 5 or 6 big plaques in major arteries, your average risk of dying is like 5 times that of somebody who doesn't have this stuff there to be imaged. And if you're one of those people with the 5 to 6 big plaques in major arteries, there's actually nothing you can do to get rid of them. That's it, you're stuck. So don't be that person. But the problem here is if you are that person, you're stuck being that person, because if you do long-term statin use, or if you do the lifestyle stuff, if you go look at the meta-analyses like these two here, dozens of studies, thousands of people, the average reduction in plaque over long-term use of statins or other strategies is 0. Or close to 0 makes no difference. That's 0.123% down there at the bottom. It's not good news. So you've all seen the drug ads with attractive older people dancing under trees, right? The picture is not as pretty as is made out. You can stay thin, you can cut out the red meat, maintain your physical fitness, quit smoking. Don't start smoking in the first place. Watch your LDL cholesterol, right, religiously. People are taught to do that now. Take your statins, talk to your doctor. But this will only slow your atherosclerosis. It's going to, on average, there will be no reduction in the plaque you have built up over a lifetime, and it's that plaque that will kill you. Back to the point again. Repair Biotechnologies has a therapy that can reliably, sizably reduce atherosclerotic plaque and do it rapidly. So this is a bit of mouse data to start with, and then I'll go back to talking about fluffy stuff again. So these are LDLR knockout mice. They're a model of a human condition in which people have very, very accelerated atherosclerosis. LDL receptor allows uptake of cholesterol from the bloodstream in the liver, and if you're not doing that, you have a lot of cholesterol in your bloodstream. Now, we're reducing plaque volume by 17% in 6 weeks of treatment. Now, back to the earlier slide I showed you again, 18 plus months of statins, average 0% reduction. So we aim to do something like this in people, bring the result from mice to humans. So how do we do this, you might ask? This is an important question. So to answer that, I have to talk a little bit about cholesterol, sorry. So cholesterol is vital to cell function, but at the same point in time, unmodified, normal, straight cholesterol floating around in your cells is actually toxic. You can't have a lot of it. But cells are very limited as to what they can do. Your body doesn't break down cholesterol. It creates it and mostly in the liver, ships it to where it's needed. It's a circulatory system of cholesterol flowing around. But the problem here is that you only have so much capacity to make it safe. So if a cell is sitting in a lake of cholesterol, it has nothing else, it can do nothing but take that cholesterol up, run out of capacity to esterify it, and now it has a lot of free cholesterol floating around, and that cell will die, or it will become dysfunctional. And this is what's going on in your liver if you're obese or you're old. Your ability to properly manage cholesterol is degraded. So what we do is we specifically target this free cholesterol that's floating around in cells, being toxic, and we get rid of only that cholesterol. This is an undruggable target, unfortunately. You can't do this with small molecules. You can't send a small molecule in the body to adapt a non-existent let's degrade cholesterol process. And you also, as Oki said, you can't just overpower the problem with standard issue cyclodextrins or similar binding proteins, because any therapy that could get rid of as much of the cholesterol as you want will also tear cells to pieces, break down their membranes, and kill you. So don't do that. So what do we do instead? So it turns out that there are human proteins in the body that if you put them together, they will break down only free cholesterol selectively. If you put them into a cell and there's no free cholesterol there, they will do nothing. But these proteins are not expressed together in cells pretty much anywhere in your body, and certainly not in the liver. So what we did, we've selected these, we turned them into an optimized fusion protein to get all of them in the same place, pulling in the same direction. Then we use messenger RNA to encode this protein. You manufacture this stuff synthetically, and so long as it ends up in a cell, it will cause expression of this protein for a period of time. So then we deliver this mRNA to cells using lipid nanoparticles. This is basically the same technology as is used in COVID vaccines, except with a much better lipid nanoparticle, much less prone to producing inflammation, and much more selective. Now, the one we use at the moment is selective for the liver. So you inject it, and 99.9% of it ends up in liver cells. And that is where you want to deal with the problem, as it turns out, which is rather bizarre, because that's not actually how we started thinking about this, but that's how we think about it now. The problem here is that your liver actually turns out to have an enormous effect on the periphery of your body. As you get old or obese, you'll accelerate this process considerably when you're obese, your liver is actually becoming dysfunctional in large part because it's accumulating free cholesterol. Your liver cells are like baking in a sea of cholesterol that shouldn't be there, and they can't deal with it. And this excess free cholesterol that's inside the cells is causing them to become dysfunctional, and it causes them to die. So we deliver our lipid nanoparticle, we express the fusion protein in the liver. That fusion protein clears out the excess free cholesterol, and this has profound effects throughout the body. And what we're demonstrating here is that this dysfunction in the liver is not like, of academic interest to your liver, just your liver. It's not only applicable to people with liver disease. When your liver goes south, the whole of your body goes south. So when we treat the liver in this way, we see dramatic reductions in blood lipids. We see reverse cholesterol transport picking up. We see a dramatic reduction in markers of inflammation. And there's a bunch of things that I can't talk to you about at all because we're busy patenting and thinking about whether they're real, that sort of thing. But there's many more mechanisms likely involved in this than the ones I outlined here before. We believe that the major components are reverse cholesterol transport improvement and the reductions in inflammation. Now, in the context of reverse cholesterol transport, I should say many people have tried bringing therapies to the clinic that improve reverse cholesterol transport to drag cholesterol back out of the periphery, and in particular out of the plaques in your arteries back to the liver. But the cholesterol that's actually being transported, if you go dig up a bunch of HDL particles and split them open and see what's in there, you'll see there's something like 50 or 80% of that is free cholesterol. So if you dial up reverse cholesterol transport, you're getting a bunch of extra free cholesterol in the liver, and that's probably actually sabotaging your efforts to improve the periphery, because now the liver's screwed up, and so more atherosclerosis in the periphery. You don't come out ahead. But in our case, all that free cholesterol that comes back to the liver is getting broken down. So yay. So we own this space, roughly. So far as we know, nobody in academia is actually working on this anymore. We licensed the the core patent from a research team in the University of South Alabama, and the inventor actually presented at one of the SENS conferences back in the day, and Aubrey introduced him to us, which is how this whole deal actually ended up happening. So there is that. And since then, we were the first to put this into animals and show that it's safe and show that it works. And we've built out a big patent tree since then, and it's still growing. We're planning on another patent this year if things pan out with one of the things that I couldn't talk to you about. So progress. Now, in terms of timeline to the clinic, so our idea is to aim for a first human trial, let's say early 2026, if the long poles all work out. And so, of course, we're raising funding for this at the moment. The idea is to go in, because it really doesn't matter for us how your plaques happen. If you have plaques, we can get rid of them. So we would like to go in first with a rare disease, because the FDA gives you a much easier time of it. You have the option of fast-track approval. You may do just one trial and get approved if your effectiveness is sufficiently obvious in the patients you're working with. So we're going to work with familial hypercholesterolemia, FH. And in particular, we're going to target initially homozygous familial hypercholesterolemia. These patients have lost the function in the LDLR gene, like the mice I showed you earlier, and they suffer for it. The people who have near complete loss of function, because both copies are broken, these people live into their 30s, and then they die of dramatically accelerated atherosclerosis, and their only treatment option is apheresis. Statins don't work on them, PCSK9 inhibitors don't work on them. You can't reduce the blood lipids in any meaningful way other than by sticking them in an apheresis device and filtering the skunk out of the blood. And you can spend $100,000 a year on getting this treatment every 2 weeks, and you're still going to die in your 30s or your 40s. And then, of course, after that, if we get an approval, off-label use for severe atherosclerosis. If you're somebody who is going to die in the next year or two because you have unstable plaque in your body, that's the story. There's nothing that can be done in the current standard of care because that plaque is there, and none of the current standard of care is going to adjust the amount of fat in your plaques fast enough to stop you dying from it. And that's an enormous market, and it's necessary. But I believe that our technology delivered to the clinic has the capacity to reduce the lion's share of atherosclerosis mortality in the world. And that's important also for other reasons, because it will give a forcing function to gene therapies. We are a gene therapy. Lipid nanoparticle mRNA is a gene therapy. It got a big boost in terms of cost effectiveness in COVID. And if you have something whereby a gene therapy of this nature can treat atherosclerosis, i.e. every older person should probably be using it every now and again, you will have a tremendous incentive to, again, dramatically reduce the cost, improve the manufacturing capacity for this type of gene therapy. And we should see a broadening of its use after that, I would hope. Side effects, useful, but not the main goal, of course. The main goal is to stop 27% of humanity dying because their arteries are corroded into gunk. So next step for us this year is actually kicking off non-human primate studies. It's an important component of getting to the clinic when you have a first-in-class therapy. You've got to test in a primate. You've got to show that it's not, because a mouse is not a man is not a mouse. It's a big leap. The FDA likes to see you taking baby steps. So we're going to work with cynomolgus monkeys because they're a very standard model for toxicity, and they're also a very standard model for atherosclerosis. We work with one of the largest and most reputable non-human primate CROs in the country, in the US. And we target later this year to complete an initial safety study. And by the mid to late next year, we will actually know whether we can reverse atherosclerosis in monkeys as well as we can reverse it in mice. And at that point, thumbs up, roll right on. So an interesting point I'll make in a moment after I show you this. Let's get back to that. If you look at this Gantt chart here, it's just a look at how we're going to roll this out. We'll complete those NHP studies by late next year. And we're raising right now 18 million, as it happens, to get to that IND submission in early 2026. And as I mentioned, the nice thing about this is it doesn't matter how you got your plaque. Our therapy will reverse it regardless. So we can start with HoFH, and then gear up to atherosclerosis in the broader population as and when it becomes a good time to do that. The initial focus is let's just get this first phase 1b in patients with homozygous familial hypercholesterolemia in the bag, and then we roll on to the bigger goals. So the point I wanted to make about the non-human primate studies and their completion, Verve Therapeutics, they went public in 2019, which is obviously a whole different era in comparison to the present market, but they went public in 2019 at the point at which they only had non-human primate data at a nearly $1 billion valuation. And these guys have just another lipid-lowering therapy. It's a really nice lipid-lowering therapy. It's a good one. But they're just lowering lipids. And we know that lowering blood lipids does not reverse plaque. It doesn't. It doesn't matter whether you take statins, PCSK9 inhibitors, ANGPTL3 inhibitors, or Verve's approach. It's not going to reverse your existing plaque because your existing plaque is not very sensitive to the amount of cholesterol in your bloodstream. We will reach the same point that they did in 2019 in late 2025 with a far superior therapy. And that's just something for the investors and the audience to consider as we say again that, hey, we're raising. Now, atherosclerosis is not the only thing we do. And why stop here? You remember, we're targeting the liver, and a screwed up liver is screwing up a lot more than just your vasculature. So we've also shown that in the case of metabolic dysfunction-associated steatohepatitis, which is the new name for NASH, it used to be non-alcoholic liver disease, we've demonstrated that we can reverse the fibrosis of the disease to a greater extent, far more rapidly than the present recently approved therapy for Madrigal can do. And in the course of doing that, we demonstrated that we can also improve in these obese MASH model mice. We can improve their insulin metabolism dramatically. Therefore, we are probably a good treatment for type 2 diabetes as well. Those guys should just lose weight, frankly. In neurodegenerative disease, it's well known that many of these are characterized by disrupted cholesterol metabolism, disrupted lipid metabolism in the brain, and lipid droplets. There's a whole field just waiting to happen here to take our approach, do the research, and show that you can treat these conditions by reversing this buildup of lipids in the brain. And there's a lot more as well. There's a whole bunch of weird, rare conditions that are very unpleasant because your body has forgotten how to process cholesterol in some part of your body. We like alveolar proteinosis, where patients literally have to have their lungs washed out with detergent every so often. And we could treat these things. So there are research collaborations in our future, and we're happy to talk to anybody who'd like to come in and work with us on some of these things, as we will certainly have our hands very full with treating the whole world's atherosclerosis for the foreseeable future. So I wanted to go back to the atherosclerotic mice because it's terrible to give a presentation and only show you one piece of data. So maybe I'll show you 4 pieces of data. 5, maybe. This is a really great picture, and I'm hoping it's actually coming out here. So when we were doing a quick little time series study where you inject the mice 1 dose, and then you just let them go for a period of time and take serum samples and tissue samples and see what you see. So we had the team lay out all the samples on the stripy board you see here and take a snapshot. So this is actually true in humans as well, but serum is normally clear, blood serum. In people and mice where your LDL gene is not working, your blood is so full of gunk that the serum actually becomes opaque. And you can see that on the left there in the PBS group and the shortly after injection group. But by 96 hours, the serum has come back to being clear. No other therapy can do this in this condition. We've completely set back their serum to the ungunked state that it should be in. So this is a great picture. Now, I showed you this one already. This is, and the point I wanted to make here is that this 17% average is over 4 or 5 or 6 groups that were dosed at a wide range of doses. We aimed at sort of 0.25 to 1.5 mg per kg for these mice because in humans and mice, LNP-based mRNA therapies are sort of floating around the 0.2 to 1.0 mg per kg, depending on the therapy. and it worked for all of them. It has a very wide therapeutic window. And you can't really overdose with what we're putting in there. You can certainly overdose with lipid nanoparticles, but you can't overdose with the mRNA that we're putting in there, because if the protein doesn't find any cholesterol to degrade, it just does nothing. So to follow up on that, we stuck these mice on a treadmill. So these mice are not going to win any prizes. They're sedentary, they're fat. But the treated mice, all of them, at all of the different doses, did tremendously better on the treadmill than the control mice did. This is a huge effect. You're clearing out the narrowed arteries throughout the body, and you're enabling these mice to have a better cardiovascular function, which is exactly what you want to see. You're improving their quality of life. Now, I should also talk about one of the other mouse models, which is the ApoE null mouse model. These mice actually have sort of disrupted reverse cholesterol transport. So it's very interesting that our therapy still works in this model. It has a slightly different character of effect, but it still works. So in this study, we compared our therapy with statins, which, as you'll recall, do not reduce plaque in the slightest, but they also don't do a lot in general in the short term. So again, 6 weeks of treatment, and we dramatically reduce the amount of cholesterol, the amount of lipids that are actually in the plaque. And we synergize with statins. We're not worse or horrible for mice when you give them statins, which is great to know, because you're not going to find a human with atherosclerosis who isn't taking statins, to a first approximation. So this is a fast effect, and that reducing the fat stabilizes the plaques. It makes them much less likely to rupture. And if you take statins for a very long time, you stabilize your plaques, but you replace the fat with calcification, which is not desired, but it's more desired than having a heart attack. Now, in our case, we don't replace it with calcification. I'm not going to show you data, but in fact, we reduce calcification in plaques in mice. These models are just not very good for showing that, because very few of them have very significant calcification. But we replace the fat with collagen, so the plaques become fibrotic and stable. And this is, again, it's a much faster effect, and it's an effect that a atorvastatin doesn't give you, or indeed any other statin. So, people would line up at your door just for this. You're stabilizing your plaques. But arguably, the LDLR model is a better one for normal humans in any case, and it's certainly the better one for the homozygous familial hypercholesterolemia patients we will be treating. So these are impressive numbers. These are actually very impressive effect sizes, achieved very rapidly, achieved reliably in mice, and nobody else can do this. So a little bit about us to round off. We live in sunny upstate New York, possibly sunny right now, sunnier than Dublin, in a very low cost of operations area. We're actually based in Syracuse, near Kelsey Moody's company, for those who know Kelsey. He helped incubate us. We've raised almost 20 million to date, and 6 million of that was earlier this year as we roll into Series A. So we're looking for 18 million and have 6 million of that in hand, and certainly talking to a lot of people right now. So if you want to get in, come and chat to myself or Morad, who's also here. So I think many of you know who I am. I have a background as a patient in patient advocacy, helping funds get into the research before there was a longevity industry. I did a little bit of investing in the longevity industry, and hey, if you invest, you eventually get investor disease, which is that you look at a company and you say, that doesn't look that hard, I can do that. And then you find yourself running a company. Morad is here in the back, I think, if any of you would like to chat about the science. He is far more knowledgeable than I am on that topic, and is in fact a considerable expert on the cardiometabolic disease front. Bobby Kahn is our CMO, and he is an atherosclerosis physician whose CV is longer than my arm, and who has founded his own companies and put drugs through the FDA process for the treatment of cardiovascular disease. Bill is my co-founder from a consulting background, and fortunately handles the taxes, which I love because I hate doing corporate taxes. So, and we can't forget the team. None of this would be possible without our very talented Syracuse lab team, who are here standing outside one of the 2 restaurants that you're actually required to go to if you're ever in Syracuse, New York. So now you know 50% of the solution, and maybe I'll tell you the other one if you come ask. So these are presently the leading experts in the world on the in vivo application of therapeutic cholesterol catabolism, because we are the only people in the world who are doing this at the moment. Our victory condition, really, honestly, will be to convince Big Pharma this is what they should be doing instead of lowering blood lipids. Once Big Pharma is scrambling to produce competition and alternative ways of doing what we're doing, after we've shown efficacy in humans sometime in probably 2027, that's victory. Because there can't be enough work on atherosclerosis. There's room enough for 12 companies in this space to all be working their asses off, because there are a lot of people in the world who are going to die this year because they have plaque, and that plaque will kill them. And that is that, and I am happy to take questions.
Yeah, great job, Reason. Thank you so much for that. Yeah, and thank you in particular for giving an example of what I said earlier on about people that nobody in the room has heard of. Richard Harkonnen spoke at the conference I ran in Berlin in 2018, and I am pretty sure that nobody in the room had heard of him until that point, and here we are. So there will be people that you will hear over the next 3.5 days whom you will be hearing a great deal more of in years to come, and that is why I make sure that quite a lot of those people are present at every meeting I run. So, questions for Reason.
Thank you so much for your talk. Amazing work. 2 questions. The first one is, do your nanoparticles cross the blood-brain barrier?
These ones do not. They literally, even if they could, they don't, and they wouldn't even get there if they could, because the liver sucks them up so aggressively. It's literally 99.9%, and nothing escapes.
The second question is about, would you be able or consider electroporating the mRNA into naturally existing nanoparticles like exosomes?
We have given that some thought, and there are certainly players in the exosome field like Exograde who are thinking about, how do we get our exosomes out beyond that therapy and into something where we can go do partnerships and get people to put their payloads into these things, but it's nowhere near as mature as the existing lipid nanoparticle field. I think it's going to be a few years yet before you can go out there and find somebody to partner with where it isn't sort of a complete research project as to whether it's going to work at all.
Hey, hi, Reason. Fantastic talk. I'm super impressed and excited about where your therapy is going. In terms of sort of the delivery, as you mentioned, it's sort of the lipid nanoparticle delivery of the mRNA that will eventually be a fusion protein. Is this going to be a one-time delivery, or is this going to be sort of multiple times in sort of the treatment paradigm? And if it's multiple, do you think there will eventually be some sort of immune response? And then are you considering other delivery? I think you might have just answered that, but other delivery modalities as well.
So sort of
and I'm going to have another question. But.
Sort of in reverse order. We've worked with AAV, but high-dose AAV, especially if it's going to the liver, is sort of verboten right now. No investor will give you the time of day if you're doing that, whether or not it's going to work. So lipid nanoparticles are actually used in therapies at the moment where you're going to be dosing effectively indefinitely. The toxicity problem is definitely a problem for some varieties of lipid nanoparticle, and some are limited to what you can do because you're going to get the lipid, the main ionizable lipid will accumulate in the liver faster than the liver can get rid of it, but this is not the case for the one we work with. The actual therapeutic sort of dosage schedule we're looking at is something like 6 to 8 weeks once weekly, and then you take stock and come back later. We actually have a study that we're just kicking off now where we're going to just do the mice for 6 weeks, leave them alone for a month, and then come back and do it again. Because in principle, you can just keep doing this and get additive effects as you go forward. But in practice, dosage is a hard problem, right? As you well know from your work. So it's a work in progress. But that should hopefully answer your question.
And then just one last one. When you say that's like a fusion protein of a couple proteins, are these proteins that are canonically sort of inside the cell, and so you would need to kind of deliver it into the cell to make it work?
Yes, this all happens inside the cell because that's where the free cholesterol is the problem.
So, last question.
That was quite inspiring. Thank you. I'm just wondering to what degree plasmapheresis or simple plasma donation might couple in with this somehow.
Judging from the experience of the homozygous familial hypercholesterolemia patients, it doesn't help that much, unfortunately. You can't plasmapheresis your way into having a lower LDL cholesterol, and even if you did, it only reduces and slows. It doesn't reverse.
All right, thank you.